वेलकम बैक डियर स्टूडेंट्स। आज हम डिस्कस करने जा रहे हैं स्पोरोफाइट और ब्रायोफाइट्स। सो इन द प्रीवियस पार्ट वी डिस्कस्ड फर्टिलाइजेशन। हमें पता है जो गेमेटोफाइट्स है ब्रायोफाइट्स में, दे प्रोड्यूस मेल एंड फीमेल गेमेट्स एंड दीज मेल एंड फीमेल गेमेट्स विद द हेल्प ऑफ वाटर दे फ्यूज and result in the formation of a fertilized egg or zygote, and this zygote it is regarded as the first cell of sporophytic generation. जो zygote है it is the first cell of sporophytic generation. Now what happens to zygote? ये जो zygote है it divides immediately without undergoing any resting period. It divides and develops into an embryo, and because of the formation of embryo in bryophytes, they have been placed along with tracheophytes. Tracheophytes are those plants which possess the vascular tissues. They have been placed along with tracheophytes in embryophyta. इनको हम embryophytes भी बोल सकते हैं because they show embryo formation. The embryo it later on develops to form the sporophyte. Okay, जो embryo होता है it develops further and forms the sporophyte. Now this sporophyte in case of bryophytes and other plants it is concerned with the production of haploid spores. Sporophytes spores बनाते हैं, gametophytes gametes बनाते हैं. Okay, clear? So we have the zygote that divides to form the embryo, and embryo divides further to form the sporophyte, and this sporophyte it produces the spores with the help of meiosis. नाउ द वॉल ऑफ वेंटर, जो वेंटर का वॉल होता है, मैंने आपको पहले ही बोला व्हाट इज वेंटर. वेंटर इज इट इज़ अ पार्ट ऑफ आर्चगोनियम. ओके, द वॉल ऑफ वेंटर it divides, it develops and forms a protective covering. It develops here this wall, this layer which is known as calyptra. It is basically a product of venter wall. The wall of venter it divides and forms a protective covering, and that protective covering is known as calyptra. So the sporophyte, or you can say sporogonium, it is covered by a protective covering that is known as calyptra, and this calyptra develops from the wall of venter. In case of Markensia, you can find another protective covering that is known as pseudoperianth, and this pseudoperianth it develops from the cells of the sporophyte. Here at this point the cells of sporophyte they elongate and result in the formation of second protective covering which is known as pseudoperianth. Now the sporophyte is completely developed. Okay, this sporophyte it is differentiated into foot, seta and capsule. Here you can see in the image, this is foot. It is a bulbous region, it is a swollen region, and it is embedded in the gametophyte. Then we have seta. This is small structure, a stalk like structure which raises the capsule. Capsule को ऊपर की ओर ले जाता है. Okay, so the second part is seta, and the third one is capsule. Capsule is that region of the sporophyte which produces the spores. Here you can see this is the anatomy of the sporophyte. Here anatomy, and this is the morphological. This image shows the morphological features of sporophyte. This is the gametophyte. Okay, and this is the sporophyte. ये जो sporophyte होता है it has three parts: foot which you cannot see in this image, okay, that is embedded in the thallus of the gametophyte. Then we have seta, this region, and finally capsule, this region. This region produces the spores. The sporophytes they are completely dependent on the gametophytes for water and mineral supply, and in most of the cases they are either partly or wholly dependent on the gametophyte for organic nutrition. क्योंकि कुछ स्पोरोफाइट्स में देखा गया है उनके अपने भी स्टोमेटा है, उनके सेल्स में क्लोरोफिल पाया गया. दैट इज़ वाई इट इज़ बिलीव्ड दैट दे आर either partly or wholly dependent upon the gametophytes for organic nutrition. Clear? Once again I will tell you, the gametophytes they are completely dependent upon the sporophytes are completely dependent upon gametophytes for water and mineral supply, and in most cases they are partly or wholly dependent on the gametophytes for organic nutrition. The sporophytes, they remain attached to the gametophytic plant body throughout their life cycle. Okay, throughout their life, the sporophytes remain attached to the gametophytes. You know that the sporophytes are distinguishable into foot, seta, and capsule. In the Rixia, however, the sporophyte is represented by capsule only. This is very much important. In majority of the bryophytes, the sporophyte is represented by foot, seta, and capsule. In Rixia, however, the sporophyte is represented by capsule only. There is no foot and seta. Now we'll talk about the different structures of capsule. First of all, foot. Foot is a bulbous structure which is embedded in the gametophyte, and the function of foot is it fixes the sporophyte to the gametophyte. It attaches sporophyte to the gametophyte. The second one is seta. The main function of seta is it raises the capsule. It raises the capsule above. Okay. And this seta, it is small in Spherocarpus, which is a bryophyte, but somewhat longer in Markensia. It is slightly longer in Markensia. And the seta, in case of Anthoceras, it is meristematic and it progressively differentiates into capsule. These are some important details about seta. Seta is the structure which is meant for raising the capsule. The size of seta is small in Spherocarpus. It is somewhat longer in Markensia, and most importantly, in case of Anthoceras, the seta is meristematic, and the cells continuously divide, and the seta progressively differentiates to form the capsule. Now, what is capsule? Capsule, it contains 1 to 2 cell jacket enclosing a mass of sporogenous cells. However, in case of Anthoceras and mosses, the wall of capsule, it is not 1 to 2 layered, it is many layered, it is multi-layered. Okay, so capsule, it has a mass of sporogenous cells which is enclosed by 1 to 2 cell jacket. However, in case of Anthoceras and mosses, it may be multi-layered, it is multi-layered. In Rixia, most of the sporogenous cells, they behave as spore mother cells. Almost all the sporogenous cells in the capsule of Rixia, they behave as spore mother cells. There are no other cells present in the capsule of Rixia. In Spherocarpus, along with the spore mother cells, there are some other cells which are known as nurse cells. They are sterile. They do not result in the formation of spores. Yaad rakhna. In Markensia, along with spore mother cells, there are some other cells which are known as elaters. Okay, and these elaters, they are diploid. Remember, in the capsule of Markensia, there are two types of cells: spore mother cells and elater mother cells. While spore mother cells undergo meiosis and result in the formation of spores, the elater mother cells, they are diploid. Okay, they develop hygroscopic thickenings, and the main function of these elaters, they help in dehiscence of the capsule and the dispersal of the spores. Yaad rakhna, this is very much important. In Markensia, inside the capsule, there are two types of cells. One is known as spore mother cells, and second one is known as elater mother cells. The spore mother cells undergo meiosis and result in the formation of spores, while as the elater mother cells, they don't undergo meiosis. Okay, they result in the formation of elaters, which are long spindle shaped cells, and these long spindle shaped cells, they develop hygroscopic thickenings in their walls, and the function of these hygroscopic thickenings is they cause the dehiscence of capsule and the dispersal of spores. In Anthoceras, however, there are no elaters. Instead, it contains multicellular structures. It contains multicellular structures which are known as pseudoelaters. These are known as pseudoelaters because in these elaters there are no hygroscopic thickenings. याद रख लीजिए, जो elater mother cells होती हैं in case of Anthoceras, this is an elater mother cell, it divides. Okay, it divides a few times and results in the formation of multicelled branched or unbranched structures, and these multicelled branched or unbranched structures, they are known as pseudoelaters. These pseudoelaters, they do not possess the hygroscopic thickness. The capsules of Anthoceras and mosses, they possess a central sterile tissue which is known as columella. This is very much important. The capsules of Anthoceras and mosses, they possess a central sterile tissue which is known as columella. Now the formation of spores, how spore formation takes place. The sporogenous cells which are present inside the capsule, they undergo meiosis to form tetrahedrous of spores. Okay, they are in the form of 4, they are in the groups of 4. When the spore mother cells undergo meiosis, there is the formation of tetraheads of spores, a group of 4 spores having tetrahedral symmetry. Okay, now the dehiscence of capsule, how capsule dehiscence occurs. Capsule dehiscence means breaking up the wall of capsule so that the spores can liberate. Depending upon the species, the dehiscence of capsule differs. It is irregular in Markensia. Markensia में जो capsule की dehiscence होती है, that is irregular. But in case of Anthoceras and several mosses, the capsule dehisces along a predetermined line of dehiscence. That means जो capsule होता है उसमें एक खास रीजन होता है जहाँ पे dehiscence स्टार्ट होती है. क्लियर? सो मार्केंशिया में कैप्सूल की dehiscence irregular है, but in case of Anthoceras and mosses, the capsule dehiscence, it is along a predetermined line of dehiscence. And accordingly, there are 2 groups of mosses: Cleistocarpus mosses. Cleistocarpus mosses are those mosses where capsule dehisces irregularly, while as Stegocarpus mosses are those mosses where the capsule dehisces along a line of dehiscence. Now, the capsule is dehisced, the spores are liberated. What are these spores and what is the function of these spores? The spores, they represent the first cells of gametophytic generation. They germinate to produce the gametophyte either directly or through a juvenile filamentous stage which is known as protonema. In case of mosses, you will find that the spore, it germinates and form a filamentous stage which is known as protonema, and this protonema, it produces upright gametophores. Here you can see this is the spore. It is a haploid structure. It divides in case of mosses and forms a filamentous structure, and this filamentous structure is known as protonema. ये जो protonema होता है, it gives rise to upright gametophores in case of mosses. Okay, here in this image you can see these are the sporophytes of different species. This represents the sporophyte of Riccia. This is the sporophyte of Markensia. This is the sporophyte of Anthoceras, and this is the sporophyte of Funaria. You can clearly see in this image the central sterile region in the sporophyte of Anthoceras and the Funaria. Okay, this central sterile region is known as columella. One important point you will remember here is in case of this Anthoceras, the sporophytes, they are just like horns. That is why they are known as hornworts. You can see in the image, these are the sporophytes, okay, of Anthoceras, and these sporophytes of Anthoceras, they because of their horn-like appearance, that is why this group of bryophytes is known as Anthoceratopsida. Okay, hornworts, Anthoceratopsida. So this is all about the sporophyte of bryophytes. See you soon with a new lesson. Till then, take care.